Hi, everybody. Looks like we have a pretty full house today. So I want to say hello and welcome everybody to today's EpiCypher webinar. My name is Emily Madden, and I'm happy to be here on behalf of EpiCypher. And we are a company run by scientists for scientists to empower your research with cutting-edge epigenomic kits, reagents, and services, including our recently launched Fiber-seq assay. I'm thrilled to introduce our guest speaker, Dr. Zach Freeman, who is an associate professor at the for laboratory and animal medicine at the University of Michigan. He's going to share how his lab is using EpiCypher's Fiber-seq assay to gain a complete picture of transgene integration and regulation using combination of PacBio's PureTarget library prep approach with our Fiber-seq assay. This allows us a lab to both increase throughput and maximize on-target sequencing to resolve gene-edited loci without amplification. If you have questions during this time, please submit them at any point in the chat, and we'll get to them all of them or as many as we can after the talk. And so with that, please join me in welcoming Dr. Freeman.
Thank you very much. Thanks for the opportunity to talk. So as Emily mentioned today, I'm going to be talking about some of our newest assays where we're leveraging both PureTarget and Fiber-seq together to characterize novel genome engineered models. Our lab is focused on making genetically engineered animal models for people's research. And so we're a core facility where investigators will come to us and request a specific animal model to be made, and we will provide them end-to-end services to deliver this model for them to use for their research. And we're really striving to make the best models and also those that are highly relevant for modeling human disease. And to do this, we leverage CRISPR-Cas9. And so our general strategy involves using CRISPR with a guide RNA and some sort of DNA donor that we can use to change the genome of the mouse. And when we do this, we deliver it into single-cell mouse zygotes, as depicted here, using most often pronuclear injection. This then will yield us animals as pictured in green here, where they have these genetic edits that we've made, and then we can carry these forward to genetically validate them and deliver to research investigators. So you can imagine that this is a long process that's very complicated and involved. And at the end of this, we really need to be able to deliver a high quality product to our investigators that has a very clear genomic status so that they can then carry it forward with confidence into their research. So why do we use animal models, and what does CRISPR let us do? It lets us create very specific and highly translationally relevant patient models. And so this is from a recent paper from a lab here at the University of Michigan, where they were studying Dravet syndrome, which is a rare variant disease in children. And they made side by side a iPSC model and a mouse model. And what they did is they took a specific patient mutation, this SCN1B 265C to T change, and they could engineer that into the mouse model. And by doing so, this allowed them to directly compare phenotypes in the mouse model with those from the patient and understand more of the basic mechanisms that are driving what's going on in that patient, which then can be used later to develop therapies. So these are incredibly powerful systems that really are helping to push forward a lot of the science and benefit patients. And so, as I mentioned, we primarily use CRISPR-Cas9 for this, which is really a very powerful genome engineering system. The basic idea of how CRISPR works is that you have the Cas9 protein, which is depicted here in purple, and a guide RNA sequence, which is in pink. And the guide RNA sequence is designed so that it's specific for whatever locus in the genome you want. And CRISPR will scan the genome until it finds this sequence in the context of a PAM motif. And when it does, it will make a double-stranded DNA break. So CRISPR is really just a molecular scissors that allows us to make a very targeted break in the genome. And when we do this, there are two possible outcomes that can happen downstream of this cut. And these all revolve around endogenous repair mechanisms. So when you have a double-stranded break, the cell wants to fix it immediately because it's very problematic. And so the first path, and the most common path, is that the cell will do non-homologous end joining, where it will either create small deletions or insertions that we call indels that just repair the DNA as fast as possible to get rid of the double-stranded break. And this can lead to stop codons and all sorts of unintended alterations when this happens. The other outcome is that you can have homology-directed repair. And the idea here is that if you provide some DNA template that has homology arms to where you would like to make your insertion, this template can then be used to repair the break, but also insert your DNA of interest. And we are most often hoping to get this correct repair that will happen where we can insert our region of interest. But there can be many different complications that happen where we can get off-target repair. Non-homologous end joining happens the majority of the time in cells, and so it's much more frequent that we get indels compared to homology-directed repair. But you can also imagine that all of these different outcomes are unpredictable and can be quite complex to characterize by traditional methods. And so, it's very hard to make an assay that can easily identify these changes and help us sift through them to find these correct repairs that we really are looking for. Another complicating factor when we do this is that we work in mouse zygotes or rat zygotes, which are rapidly dividing. And so when we deliver the Cas9 enzyme, the Cas9 enzyme will stay active throughout cell divisions until it is no longer, the protein is no longer around. And we generally think of this as about 4 cell divisions. So when we reach a 16-cell, 8 to 16-cell zygote, the CRISPR is no longer active. So in that initial cell, we will have 2 copies of our gene of interest where we're trying to make an edit. And we can have 1 copy where no editing is happening, and then a second copy where an indel gets created as pictured in red here. That cell will then divide, and each cell will get 1 copy of each of those genes. And when that happens, the CRISPR can then cut either of those 2 alleles that has not been edited yet. And when it does, you can get another unique indel that happens. The red alleles here are protected from CRISPR activity because the guide site is no longer there. But now we have 3 different alleles present. And this can happen at each successive step until the Cas9 is no longer active. And so this can really complicate the characterization of these because you can have alleles that are edited correctly at a frequency of 5%, and that can be biologically meaningful. Yeah, you have to be able to detect that via sequencing. So mosaicism is really another complicating factor. So this is an example of what mosaicism looks like practically. And in this experiment, we are doing PCR and long-read amplicon sequencing. We've taken mouse zygotes and we've injected CRISPR with a guide RNA for a target region as pictured in red. And we have coverage tracks from 9 different embryos here. The top embryo is a wild type embryo that's not been treated with CRISPR. And you can see that throughout the entire coverage track, we have 100% coverage at every position. And then the bottom 8 strips, each one represents an independent embryo that was injected. So each one had the guide injected, and then it developed into a blastocyst that we picked up for sequencing. And you can see centered over the guide that we have activity in most of these embryos that have developed, but the pattern of these mutations is different. So there are unique patterns and then also unique frequencies. Some of these we have very large deletions that are, 1,000 base pairs long. Some of them that are much smaller. And so we really need assays that can be able to detect this, because finding that, correctly inserted transgene or DNA that we're trying to make can really be confounded by this. Another thing that happens quite frequently when we're doing these events is that we can get what we call complex rearrangements. And ideally, we're going to make 1 cut as depicted here with this guide. We're going to provide a donor that's going to introduce this cDNA of interest. In this case, it's a human ACE2, so that this human ACE2 will be driven by the mouse ACE2 promoter. And afterwards, you would then end up with this construct where you have, a complete insertion of exactly what you want. And you can design PCR primers to cover each of these regions and then screen these and ideally be able to tell that they're correct. This is from a published paper where they did all of the genotyping using traditional PCR primers, and they thought that they identified founders that were correct by each of these different PCR assays. but then they did long-read sequencing to confirm it. And when they did, what they found was that they had a head-to-tail concatemer where actually 3 copies of the transgene had gone in a row. And so even though the PCR said that this animal was validated correctly and it was all edited fine, they actually saw that there were multiple copies in a row. You can imagine that this is not a desirable outcome and could really impact how well this model system works. So when we're generating these models, they're quite expensive to make. They're very time consuming. And we really need to make sure that we can generate accurate characterization to ensure that we're getting the editing we want and that this is relevant to the human context that we're trying to model. So ideally, we'd have an assay that can detect any edit without knowing what it's going to be. We also need to be able to detect very low frequency variants. We've had correct edited alleles at around 5% where they will germline transmit to the next generation, meaning that they're real and we can then make a line out of them. And we really need to be scalable and cost effective. Our lab is characterizing hundreds of mice a month, and we need to be able to do this in a high throughput fashion and also have it not add a lot of cost. To make a model costs somewhere between $10,000 and $15,000. And so no one really wants to spend another $15,000 to genotype them to find those positive animals. And so to do this, we have been using PacBio's PureTarget library with custom guide RNAs to do CRISPR sequencing. So CRISPR sequencing is the idea that we can use guides to target areas we want to sequence, and we can then cut that area out and do sequencing on it and get a targeted enrichment. This works by first we dephosphorylate the DNA so that we get rid of all the free phosphates. Then we use our guide RNAs to cut the area we want to sequence. So this is outside of where our guide RNAs were used to make the model. We will go usually several kb downstream. And when we cut the DNA, that then frees up phosphates that we can use DA tailing and then do our downstream barcoding and library prep to be able to use for sequencing. So on a PacBio Vega, which is what we have, you can do 48 samples in a single flow cell, and on a Revit, you can do up to 96. So this allows for really scalable sequencing of a lot of samples in a targeted way. The original PureTarget assay was developed for human repeat expansion diseases to be able to characterize those. And they've recently come out with a kit that allows you to use custom guides. So what we've been doing is we design guides for our region of interest, and then we use those with the library prep kit to characterize that region. And so one example that we started with is the mouse Rosa 26 locus. So the mouse Rosa 26 is very commonly used as a safe harbor to put transgene insertions in there. And so we generally put in a promoter, which is the CAG here, with some sort of cassette to control expression. We can put in a cDNA into a multiple cloning site and then use this system to make a lot of different models that have just varying cDNAs. And so we designed guide RNAs that sat outside of the homology arms so that we could sequence essentially any Rosa 26 model using the same assay. Our lab makes about 15 to 20 of these a year, and so it really allows us to test these in a very high throughput fashion. The other advantage is that it also works on wild type DNA. So you can run this on control samples, look at what a wild type sample looks like, and then see how much coverage you're getting. So that was our first step, is we did this on wild type samples. So this is an example of a wild type animal that's on a mixed genetic background. So it has, mice are usually inbred, meaning that every allele they have is the same. But in this animal, it has alleles from 2 different strains. And so if you, first off, you can see that we have 678x coverage. So we have very high coverage in this region of interest. You can see that the reads stop very abruptly right where the guide RNA sites are. And we can also see that we can phase the reads. So we have reads that align perfectly to the wild type sequence here, which is the black 6J strain. And then we also have reads where we can see single nucleotide or small variations that are from the other strain differences from the black 6J reference. And so we can get very high coverage and resolve single nucleotide variations between these. We've done this assay now with many different numbers of samples per flow cell, and we've seen that you get high coverage consistently, really even up to 48 samples on a flow cell. So here, this is from 2 different runs, and we're looking at the coverage per number of reads for an individual sample. Each dot represents a sample, and we can see that if we run 12 samples or 32 samples on a flow cell, we can get on average 150x coverage, and sometimes 500 to, we've seen even 1000x coverage across the region of interest. So this really gives us scalable, high depth coverage on long read. And just by comparison, if we were doing whole genome, we would get 25x coverage on 1 sample out of the same flow cell. So this is really a large difference in coverage we're getting. So we wanted to test this and see if we used it on a known transgenic allele, what would it look like? What would the coverage look like? And so this is from an example where we had made transgene insertion into rows of 26. And so on the top here is a coverage track, a bed file that's just showing you the structure. And so we have a homology arm. This is the promoter sequence. Here's our cDNA. And these are all the different elements within our transgene. And we can see here that we get, a good number of reads across this where we have full length reads demonstrating that there's no variation in the sequence of interest. So then if we focus, we can look even closer into our different regulatory elements within this construct and see that we have a template perfect insertion and there's no errors present within this. And so we can then take it forward and validate it. So this is really powerful because in our normal workflow, we would have to do at least 3 different PCRs to cover across this region. And so we have a standard set of PCR assays that we use where in red is what we use for the left homology arm. So these primers will amplify to make sure that we have the insertion of the promoter. We have in blue universal insert primers that we can use to make sure that the cDNA size of the correct cDNA is there. And then we have primers in green, which we can use for the other homology arm. These PCRs are large and can be rather complex. The left arm has CAG in it, which is very repeat rich. And there's also a lot of GC content within the rows of 26 promoter that makes these PCRs challenging. And so our normal workflow without PureTarget, we would do PCRs for each of these. And so I just want to show a few examples of those. So here, this is the left arm PCR from a series of animals. And for each gel image, we have a ladder that has size markers for controls. And we can see on the top gel, animal 52 has a positive band, which is the correct size for this insertion. Animal 99 on the bottom gel, I've highlighted because it does not have a correct band for the homology arm. But we're going to follow that animal through because it helps to really highlight the utility of PureTarget. So when we look at the insert sequence, we can see that in both animal 52 and animal 99, they have a correct insert sequence. So we do sometimes see animals where they have the insert sequence, but it's randomly integrated. So this insert assay will pick that up, but it's not sensitive for a target integration. And then when we look at the right homology arm PCR, we can see that both animal 52 and 99 have a correct insertion of the right size. So animal 52, we would carry forward and say it's correctly edited, and we could use that downstream. But in animal 99, we would have previously said this is not correctly edited. So in an experiment, when we make these models, sometimes you only end up with 1 or 2 animals that are correctly edited. And so if we miss an animal like this, that might mean we have to go back and start the project over again. And so it's very costly to not find these. So we were curious as to if we did PureTarget, would these animals have correct sequence and that we're just missing it with the PCR assay? And so we did PureTarget on both of these animals. Again, we have a bed file showing the structure. Animal 52 is the one that was correct, and we can see that it has full length sequence that's correct without any errors. And in animal 99, we actually have the same, we see reads that support the same thing. So even though by PCR, animal 99 was not a positive, by PureTarget it was, which shows us that we are potentially missing animals because of the issues with the sensitivity of PCR. I also talked about earlier how we see complex rearrangements happen where we can get transgene integration that is not template perfect. And so this is another example where we're in rosa 26. This insertion was supposed to be around 16 kb, and we would expect that we should see reads that have a perfect template repair. But you can see immediately here, though, we have a lot of reads that have these rainbow reads, which are soft clipped. And so these are reads where they're aligning to multiple places in the reference. The reference sequence here is the correctly edited transgene allele. So if we have a correct read, it should not have any of these mutations. But here we can see that there's all of these reads. Another thing about these reads is that they actually were very long. And so we took these reads and we blasted them against the original sequence to see what might be going on. And so this is the dot plot as a result from that. So on the y-axis here, we have the design sequence. And then on the x-axis, we have one of the reads that goes across this region. And so in a normal correct edit, we should see a diagonal line that starts in the bottom left corner and goes all the way to the upper right corner. And there should be no breaks within this line. And here we can see that there's multiple breaks in the line, and there's also overlap in the regions of sequence on the x-axis. So this suggests that there's at least 2 copies concatenarized in this insertion. By PCR, this animal looked like it was completely right, like that we had a correct insertion. But when we do this PureTarget assay, we can see that, in fact, that there's a concatenator present in the integration site. We can also detect mosaicism. So this is from a different genomic locus, and we have 2 samples. The sample on top is one that was edited with a guide, and the sample on the bottom is one that wasn't edited. And you can see in the top here at the cut site that we can see 2 different edited alleles and 1 wild type allele that are all detected. And so we can see this mosaicism and see down to even very low frequency events on the PureTarget. So far I've talked about genomic validation, which is a lot of what we've been chasing, but we are also thinking about epigenetic validation of these engineered constructs, because we know that if you make certain genetic alterations, you might cause unintended epigenetic changes. So you could have a structurally correct repair that disrupts some sort of epigenetic function. We are also often introducing promoters and highly complex regulatory elements that likely have impact on other endogenous gene elements. We work in many different background strains, and we know that they can have sequence variation and possible epigenetic variation. So for us, really an ideal assay would allow us to look at both the genomic and the epigenomic changes at the same time. One of the benefits of long-read sequencing is that you can do this. We can detect both the genetic elements and the epigenetic elements. And so we've been starting to think about how can we develop assays to do this. And this is just one example. So this top sample is another mixed background mouse where it has 2 different background strains. And so you can see here that this top set of reads aligns perfectly to the reference because these are from the black 6 allele, and then this bottom strain is from another allele. This animal below here is completely black 6, and so both copies of this gene are black 6. And in each of these examples, you can see we have more than 350x coverage by PureTarget. And so if we focus in on some of these regions and look at the 5MC patterns, we can actually see that there are changes in 5MC associated with these variations. And so here we're looking at a zoomed in region of this variation, and now we have all of the 5MC patterns in red. And you can see the alleles are still phased by the mutations here. So there's a variation right there, and there's a mutation here, and there's a variation here from the reference. And we can now see that in this animal, there is actually an added location of methylation that is not present in the wild type genome. So this is helpful for us to know, and we can actually use this and make sure that when we're selecting founders, we can determine that the insertion happened in the black 6 allele and not in the other allele, so that we can eliminate these possible confounding effects on the transgenes. But 5MC is just one mark of epigenetics, and we were interested in other possible marks. And so we wanted to explore Fiber-seq. So Fiber-seq is an assay that leverages M6A methylation, which is not usually present in eukaryotic genomes, to look for open promoter activity. And so the basic idea is you take your nuclei that have chromatin in them, and you use an M6A methyltransferase enzyme, which will then identify open accessible adenines and create M6A marks. And so here we can see that anywhere where the DNA is bound to proteins, whether those are chromatin or they're transcription factors, we do not get any M6A marks, but in all the open regions, we can get the M6A marks. We can then extract this DNA and read it on a PacBio sequencer, and we can see the M6A in addition to the 5MC, and then look for where the chromatin was bound or not bound. And so in one assay, we can see all genomic, 5MC, and then M6A methylation. And so we've done this with whole genome from mice that have transgenes. And so in this case, these animals have two transgenes that are in the opposite orientation. So this is the first transgene here and the second transgene. And there's two different promoters, but it's the same promoter in there twice. And so if we look at the transgenic alleles here, and we're looking at the 6MA, we can see that there's plenty of marks here present in the promoter that suggests that these promoters are open and active and they're being expressed. And if we look at the non-promoter regions, we can see that there's not any marks. So this is really quite powerful for us. And we've been wanting to do this for more models. The problem with whole genome is that we do not get very high coverage. And so it's very difficult to completely resolve where the promoters are. And we can only do one sample at a time. So it's very expensive. And so we wondered if we could combine Fiber-seq with PureTarget to get higher depth coverage and a better sense of the landscape of chromatin in some of these models that we're making. And so to do this, we took the PureTarget assay with our guides that we designed for in our ROSA 26 assay. And then we used the Cutana HIA5, which is the methyl 6A transferase from EpiCypher, to perform Fiber-seq at the same time. So really the only difference in our workflow is that we had to extract nuclei, use the Fiber-seq enzyme on those nuclei, and then go into our downstream high molecular weight DNA extraction for the rest of the assay. So it really just added one step to our current protocol. And so just as a first pass, we wanted to know if we look in this ROSA locus in wild type samples where we've characterized it genomically very well, do we see any, can we see evidence of chromatin stenciling? And so within this, there is a known promoter that has two different promoter peaks as depicted in the bed track on the top here. So we're going in the antisense direction. The top sample here is without any HIA5 treatment, and you can see that there's no marks or very, basically background level of marks here in nothing that falls within the promoter. So then we tried two different HIA5 enzymes. We got one from the Andrew Strigakis lab, who was the original author of the Fiber-seq paper. And then we also got the EpiCypher Cutana HIA5, and we treated the same samples that got no treatment and looked for marks within the promoter. And so you can see here very clearly in both of these samples that we get good stenciling within the promoters. We can actually narrow the area to see more specifically where most of the activity is being driven from, and we can get a good idea of where the activity is happening and what it looks like in these wild type samples. So we got good stenciling here, and we also have 5MC and all of our other long read sequencing on the same data set. So we next wanted to test samples where we knew that they would have different chromatin patterns. We first ran this on splenocytes, which is here, and you can see that we get the same pattern where we have good chromatin stenciling as we saw, but we also wanted to do it in sperm. So we work a lot with sperm because we're doing a lot of reproductive biology work to make these models, and we know that sperm DNA is highly compact. It typically has very close chromatin states, and so we figured that if we did Fiber-seq on these samples, we should be able to detect whether or not we could see any evidence of stenciling. And so when we did this, we saw exactly that. So we saw no evidence of any promoter activity within the sperm for this ROSA 26 location, which is what we would have anticipated based on our knowledge of how sperm DNA is bound. So, we were excited to see that the enzyme was working. We can identify promoters, and we can also see promoter activity that's consistent with known biotypes that should have different activity levels. So, we were really interested to try to understand in a transgene context, if we've added a promoter, how does that impact the endogenous promoter? So this diagram depicts for you what two alleles in a mouse that has a transgene would look like. So it will have one copy of the wild type allele, which is the bottom pattern. We'll just have the ROSA 26 promoter. And then it will have one copy where it has the ROSA 26 promoter with the CAG promoter, and then the rest of the downstream DNA elements that we've engineered in. And so we were interested to see first, if you look at ROSA 26 and CAG together, can you see promoter activity from both of those? So we did PureTarget on this, and I just wanted to show the overall structure of this. So we have the endogenous ROSA promoter here. This is our CAG promoter. In this region, we have our cDNA, and then the homology arms. We can see that the reads all have a perfect insertion here, and there's no sort of concatenation. And then I'm also showing here, this is the 5MC pattern across here. So we can see within the promoters, there's essentially no 5MC activity, which is as we'd expect. And then in the cDNA, there's sub 5MC and also some within ROSA. So we did Fiber-seq on the sample and then looked to see what is happening with these various promoter sequences. And so when we did that, if we focus just on the promoter, we can get a better idea of what's going on. So again, the top here is a bed file showing you the structure. So we have the ROSA 26 promoter, which extends into here. We have the CAG promoter, which consists of the CMV enhancer, the chicken beta actin promoter, which is the text isn't showing, and then a chimeric intron. If we look at the 5MC, we see very little 5MC across this region. But when we look at the Fiber-seq patterns, we can see that we're getting consistent marking within ROSA, as we've seen before. But now we can also look within the CAG promoter to see what's happening. So here we can see that we get a lot of activity within the CMV enhancer, which is what we expect because it's a very strong promoter. And then we can also see some activity within the chicken beta actin and the chimeric intron. And so we can really validate here that this promoter is open and it at least looks like it's active in the way we would anticipate that it would look. We were also interested in the other allele. And so we often have these other alleles and we don't really know if there's any impacts of having this CAG promoter on the other ROSA 26 promoter. So is there some sort of trans effect in the epigenetic state that happens when we introduce these in there? So to do this, we wanted to look at the wild type ROSA 26 promoter in an animal that has another allele with CAG and then another animal that has just a wild type allele. And so here we're looking again at the wild type ROSA 26 locus. We have a wild type animal. So this has two copies of the wild type endogenous ROSA 26. And then we have a ROSA 26 transgenic animal, but we're looking at the wild type allele. And when we do this, we can see that the pattern is really the same. There's not a lot of difference in the overall marks between these two animals, suggesting that there's not really a trans effect of introducing this very strong promoter. So in conclusion, PureTarget really allows us with custom guides to get very high depth coverage across gene edited samples in a very scalable way. We regularly do 48 samples in a flow cell and get very high depth coverage across them. This has helped us to find animals that were missing by other approaches. And so it's really increased our efficiency for our genome editing or increased the detection of this, which is helping us to make our projects more efficient. When we combine PureTarget and Fiber-seq together, we really get high depth chromatin stenciling, and we can get genomic and epigenomic validation that's cost effective and very scalable. I will also say we've so far have done this mostly in a few loci, but it really can be extended to any loci and for any application where you want to know, where you need a lot of coverage and you want to look at the epigenetic state of that locus. And so we're really starting to think now about not just genomically validating our model systems, but also epigenetically validating them so that we can really ensure they're replicate, the phenotypes in people that we're trying to model with these animals. So I just want to take a minute to thank all the people who did all the work, because without them, none of this would happen. So in our lab, Honglai Zhang and Jennifer Leo have done all of the CRISPR work, and then a lot of the work for isolating DNA and the pre-treatment for Fiber-seq. And then we are really fortunate that we work with our advanced genomics core, and they do all of our sequencing. And so we have this great partnership where we prep the samples and get them the samples, and then they do the actual sequencing for us. And so Shipper Garg does most of that. And then Xuansong Hong is our bioinformaticist who does all the analysis and the data processing downstream.
Awesome. Thank you so much for that, Dr. Freeman. That was an excellent example of how Fiber-seq can be combined with PacBio's PureTarget to rapidly assess transgenic animal models. Just a reminder to everybody, drop your questions into the question box. I see a lot of you guys are doing that. We'll get to those at the end, but right now I'm going to zoom out a little bit and share some other ways that Fiber-seq, other data that Fiber-seq provides and how it might be used in a gene editing context for you guys. So you guys should be seeing my slides. Yes, okay. So Dr. Freeman gave a great overview of how his lab uses CRISPR-Cas editing systems. So I'm just going to get a really quick, brief, broad overview again of the two major categories of editors. The first being sequence editors like CRISPR-Cas9 that was just described. These are directly modifying the DNA sequence, including everything from simple indels up to precise substitutions or large knock-ins. Another category I would be remiss not to mention, being an epigenetics company, are the epigenetic editors. So these are editors that, such as deactivated Cas9, that are fused to epigenomic components like CRAB, P300, DNMT3A. These tools modulate gene expression without altering the DNA sequence by rewriting the methylation, histone modifications, or chromatin accessibility. So regardless of which editing system that you're using, researchers are going to want to rely on several QC methods to make sure that their genes are getting expressed and the changes are being made. Dr. Freeman provided an excellent explanation for how amplicon sequencing is traditionally used to confirm that the target site was edited. And he also shared the many pitfalls that this method could have as well. For epigenomic editors, you're also going to want to look at the chromatin state. These assays tend to include ATAC-seq, whole genome bisulfite, cut and run, or CHIP. These are used to determine if the epigenomic feature you were trying to edit was actually removed as desired. But this layer is often not performed for sequence editors for many types. The other methods that you're going to look at regardless, especially if you're trying to change an expression, is qPCR, RT-PCR, RNA-seq, and Western blots. However, what I want to highlight is that most of these QC workflows are going to focus on the sequence and the resulting expression, and not necessarily the regulatory state around the edit. However, this is often an overlooked layer, but it's an important layer because it can actually make a big difference in a functional edit and one that fails. Let's see, there we go. So the other critical information needed today is obviously the epigenome. So the epigenome refers to chemical modifications on DNA and histones, as well as higher order chromatin accessibility and 3D genome folding. So these layers do not change the underlying DNA sequence, but they have a profound influence on gene expression. So importantly, the epigenome plays a major role in success or failure of genome editing. Regions of closed chromatin or heterochromatin can dramatically reduce Cas9 cutting efficiency. DNA methylation at or near target sites has been shown to inhibit Cas binding, and homology-directed repair-based knock-ins are known to perform worse in compact or silenced chromatin environments. Notably, nucleosome positioning has also played a role and can be found to affect Cas9 targeting, impacting editing efficiencies. But the reverse is also true. Gene editing can impact the epigenome. Introducing double-strand breaks initiates chromatin remodeling processes that can locally alter accessibility and histone marks. Large insertions such as transgenes can be progressively silenced through methylation accumulation, and unintended regulatory effects like silencing or activation of neighboring genes may occur even when the sequence itself is unchanged. This leads to the core challenge. Most QC workflows focus solely on sequences, focus solely on if the sequence is correct. But that doesn't tell you whether the surrounding epigenomic landscape is compatible with expression or long-term stability. So to truly validate an edit, especially for cell and gene therapy, you would want to inform on both the sequence and the regulatory state. However, I know that's more assays, right? The regulatory state around an affected site might be impacted by multiple features, and each has their own assay used to assess it. So if you're looking at a nucleosome position, you might be using MNA-seq. If you're trying to assess open or closed chromatin, that's ATAC-seq. If you're using short-read sequencing, you might be using whole genome bisulfite for methylated DNA. Obviously, using long-read would add that information. But the full picture means you'd have to run lots of assays to capture a full picture of this puzzle. And that's where we're really excited about Fiber-seq, and Dr. Freeman's presentation lets you in on, is that Fiber-seq lets you look at multiple layers of chromatin in the epigenome at the same time you're assessing the DNA sequence. So because Fiber-seq leverages long-read sequencing, you automatically get information on small and large genetic variants, along with endogenous DNA methylation, since both major long-read sequencing platforms can directly detect 5MC. However, with the addition of Fiber-seq upstream of your library prep, you can also gain a direct readout of open versus repressed chromatin, which tells you which regions are active and which are silenced. And something that wasn't alluded to before is actually the sensitivity of Fiber-seq can also allow you to infer transcription factor binding and nucleosome positioning from the same data set. So in a single experiment, you're capturing multiple features of chromatin structure and epigenomic regulation, something that normally would require several different assays. What this means for your gene editing experiments is that if you're developing a transgenic animal model or otherwise, is that Fiber-seq gives you a much more complete picture of what's actually happening after editing. You can confirm precise on-target edits and can also detect genome-wide off-target edits, including structural variants or translocations that short-read sequencing often misses. You can verify that the integrated transgenes are correctly oriented and whether or not they are active and accessible based on the epigenomic information. You could also identify unintended changes in chromatin accessibility or DNA methylation around the edited region, whether those changes come from chromatin being reassembled incorrectly after a double-strand break or from off-target effects from an epigenetic editing tool or off-target cuts that were corrected incorrectly epigenetically. And because the data are at single molecule resolution, you can also directly assess mosaicism in edited cells or founder animals. So Dr. Freeman gave a quick overview of how simple Fiber-seq is to add upstream, but just to recap this, Fiber-seq is a long-read compatible workflow, and it takes less than 2 hours to go from cells to purified genomic DNA ready for long-read library prep. The key step involved adding the HIA5 enzyme and its cofactor SAM. So these are added to purified nuclei to keep that chromatin intact. It's a short 10-minute reaction, and in that reaction, HIA5 places methylation marks on accessible adenines. After quenching this reaction, you simply purify the DNA and move into your standard library workflow. Fiber-seq is compatible with both major long-read platforms. Dr. Freeman nicely highlighted how pairing Fiber-seq with PacBio PureTarget improves throughput, but Fiber-seq is also fully compatible with direct sequencing using ONT's nanopore sequencing. Once you've got your data sequenced, you can analyze it in a couple of different ways. You can look genome-wide, looking at the overall chromatin landscape. You go by haplotype, as Dr. Freeman showed, which is especially useful for allele-specific editing. Or you can look at it at the single molecule resolution, where each molecule carries both sequence and epigenetic information. And importantly, all the tools needed for Fiber-seq data analysis are publicly available. So here's another way to look at this data. What I'm showing is a representative locus from Fiber-seq data generated from human K562 cells. On the bottom panel, you'll see a gray block. This is actually individual Fiber-seq reads. Each horizontal read across is a single read that's been aligned to this locus. So where you see white gaps is simply where one read ends and there's space between the next read being aligned. On each read, there is either red marks indicating the chromatin was accessible and labeled by HIA5, or there are black marks representing the endogenous 5MC already present on that molecule. Above, I'm showing an aggregate view of these data. The track labeled FIRE score, which stands for Fiber-seq inferred regulatory element, summarizes accessibility across all the underlying single molecules. This way to view the data is very analogous to accessibility peak calling by ATAC. And so you can actually see that the FIRE score aligns extremely well with ATAC-seq data set generated in the same cell line. So you get the same accessibility information you'd expect from ATAC-seq, but now directly tied to single molecules and combined with long-read sequencing and 5MC methylation data. So if we zoom in on this locus, the SNP214 promoter, we can see just how high a resolution the accessibility signal from Fiber-seq is. You'll notice a strong cluster of 6MA labeling right under that fire peak, which reflects the high accessibility in the promoter region. But you'll also see 6MA signal in the region on either side of this peak. That's not noise. Fiber-seq is so sensitive that it actually labels the linker DNA between nucleosomes. So this nucleosome footprinting is valuable because it lets you see how nucleosomes are positioned around a target site, helping inform what the best sequence might be for a guide RNA design. You might also notice that there's a much smaller gap or footprint directly under that fire peak. Fiber-seq can be used to detect DNA-bound proteins like RNA polymerase II or transcription factors. Because these proteins have a much smaller footprint size in otherwise accessible regions of DNA than nucleosomes. So in a single data set, you can detect nucleosome position, transcription factor, or polymerase occupancy based on the footprint size produced. So you could combine Fiber-seq with a targeted protein mapping method like cut and run if you really wanted to understand what proteins were binding in your region of interest. And so this also lets you look at the heterogeneity and how those proteins might be binding at a targeted site. Here's an example of a different locus I'm showing. It's the same Fiber-seq and ATAC-seq data, but now I've added in a CTCF cut and run track. So CTCF is a DNA binding protein involved in higher order chromatin structure, and cut and run clearly shows that CTCF is present at this locus. I've also highlighted the location of the CTCF binding motifs in this region. And you'll see that despite there being a single peak indicating CTCF is present, there are actually 3 CTCF motifs close together in this region. And neither cut and run or ATAC can tell you which motif is actually bound or whether all 3 are occupied. When we look at the single molecule Fiber-seq data underneath, the picture becomes clear. Using footprinting, areas where 6MA is missing in an otherwise accessible region, we can see that the middle motif is occupied on nearly all molecules. The right-hand motif is bound on many, but not all, and the left-hand motif is only occupied about half the time. So Fiber-seq doesn't just show accessibility, it reveals the heterogeneity in protein binding at single molecule resolution. On the right, I'm showing now a heat map summarizing this across the entire genome. We aligned all the CTCF-bound motifs based on our cut and run data. The strong red signal reflects accessible chromatin marked by 6MA, and the sharp blue line down the center, or the absence of 6MA right there, is the CTCF footprint. So this tells us that CTCF is strongly bound at these sites across the genome. We can see that there is some regular positioning on either side. These are actually the nucleosomes too, binding around that. So Fiber-seq lets you go beyond accessibility. You really understand how proteins bind and regulate the genome, both at specific sites you might be interested in looking at for your gene editing experiments, or at a global scale if you're trying to see how your gene editing has affected the global genome regulation. So you can look at the global changes in the other epigenomic features that we're interested as well. We often look at 3 key things when we're doing our Fiber-seq experiments: open chromatin, DNA methylation, and inferred nucleosome positioning around transcription start sites. So each row in these heat maps represents the same transcription start site, and the rows are ordered according to the accessibility in the first panel. So the most open and active promoters are at the top, and the least accessible are at the bottom. And what I hope you can appreciate is that accessibility and methylation track as you'd expect biologically. Highly accessible transcription start sites have very little 5MC methylation, and then about halfway down the plot, you can see a clear transition where promoters become more methylated as accessibility decreases. And then finally, on the far right, the nucleosome positioning panel is also concordant with this. The most active promoters at the top have very well-defined nucleosome structures. You can clearly see the +1 and +2 nucleosome footprints right downstream of that active transcription start site, as you would expect. This kind of genome-wide view is incredibly useful for understanding global regulatory changes in edited cells, lines, or transgenic animals. If your goal is simply to express a new gene without disturbing the broader regulatory landscape, you'd expect these heat maps to look very similar before and after editing. If they don't, Fiber-seq will show you where those things might have changed. So we've validated Fiber-seq is broadly compatible with multiple models. In our own work, we've applied Fiber-seq to multiple human cell lines in primary mouse cells. There are also published studies showing it works in yeast, Drosophila, and even maize. And we know from communications with customers, the customers are applying Fiber-seq to a lot of fun organisms and a couple emerging model systems. So if you're working with something unusual and want to confirm compatibility, please reach out to our tech support team. We're always excited to hear about how people want to use Fiber-seq. We've also tested Fiber-seq with different preservation methods. It's compatible with cryopreserved and lightly fixed nuclei, which means you can batch your samples, freeze or lightly fix them, and run Fiber-seq later when you have everything collected at once. So to bring this back to what we discussed earlier, normally validating a gene editor, a transgenic model, requires multiple separate assays. And because of this, assessing the regulatory landscape is often skipped, unless you're using an epigenomic editor. With Fiber-seq, we hope that you don't have to skip that anymore. Fiber-seq combines the assays you typically need to evaluate those first two stages of gene expression, DNA sequence and the chromatin regulatory state, into one experiment. This means less hands-on work and more complete picture of what actually happened after editing. You get higher confidence that your engineered change is correct and remains active as intended, and it isn't creating unexpected off-target effects, whether in the DNA sequence itself or in the surrounding regulatory environment. So if you're ready to get started with Fiber-seq or introduce it into your lab, we have two main ways you can get started. We recently released a Fiber-seq kit. This has everything you need to go from cells to purified DNA in under 2 hours. It contains that key HIA5 enzyme, and it also contains a manual and a bench-ready quick start card. So once you're familiar with the manual, we've got a laminated quick start protocol that you can leave at your bench for you. The other way to get started is with our services. These can be fully end-to-end, and we can handle tissue culture and isolate nuclei from your samples, run the assay, and do the sequencing and analysis. Or if you prefer to do the experiment in your own lab, but want support with sequencing and data processing, we can help you there too. All our service projects include expert guidance to help you design the strongest possible Fiber-seq experiment, plus a standardized data report and QC. You'll also have time with our bioinformatics team so you know how to interpret your results, and you can get the data analysis pipeline running in-house if that's something you would want. So with that, I'll go to the questions and see what we can answer. So I'm going to go to the bottom. So let's see. I have a question from Terrence Turner for you, Zach. Yep. This question is, how large of a transgene can you design your sequencing guides for while still obtaining the HI5 CCS reads?
Yeah, it's a great question. So with PureTarget, you can theoretically do up to 20,000 base pair reads, but you start to run into some inefficiencies in getting two guides to cut on the same strand. So it's not a sequencing problem, it's a CRISPR problem. So we're currently working on several projects where we're doing a phased design where we essentially use overlapping assays to basically tile through a larger region. So we're doing one now that's 100,000 base pairs long, and we just have several assays where they can go across that, and then we can phase the reads to make sure that we can put the whole thing together in a single assembly after we do that. So that it ends up being 6 or 7 assays that are done on the sample, basically where you do the odd tiles and the even tiles. So you can go fairly large, you just have to design more assays to get across it.
Awesome. So this might be similar to that. Is the coverage linear with the number of loci sequenced, or does the coverage quickly drop off after a number of loci are sequenced?
So we typically do one loci because we're using it to characterize an individual locus, but we've used PacBio's PureTarget panel where they have 30 loci, and the coverages, it seems to scale in that you're still getting hundreds of X coverage per target region. In this library prep, coverage is not really related to the number of preps because usually you're always putting in a very low amount of library into a large flow cell. So the issue is really it's sort of a needle in a haystack problem. And the more needles you have, you just continue to get better sequencing. We don't really know the cap on that yet. We're doing some experiments to see if you do 50 targets at once, what happens. But so far our experience is that you can add more targets and you can still get more coverage, and it's not like it's going down significantly and you're suddenly losing it.
We have a question from Sarah Kingan. She says, great talk. Can you talk a bit about how you designed your custom guides? And do you expect to eventually develop a catalog of guides that you can use for different animal models?
Sure, thanks, Sarah. Yeah, so we design our guides and then we validate that they work before we run the assay, which I think is sort of critical. So we will design a guide using we use CRISPR, which is a standard guide design tool, and we pick based off of some of the scoring metrics about how well the guides cut DNA. We also pick to minimize off targets, because unlike in vivo, when you use guides on naked DNA or even DNA with chromatin, you're going to get more cutting at the off target sites. And then critically, we validate in vitro that they actually cut. So we make a target for the guide, we cut the target, and then ensure that it's actually getting cut. And then we're also now actually using as a spike in control to make sure that we can do, like a wild type sample to get good coverage. So we are working on publishing that to say this is the method for how we're validating the guides. And then I think eventually we will put out the guides that we can use for conserved loci. So we have about half our projects where we're doing conserved loci, meaning we're just using the same guides over and over again. And then the other half, we're designing custom ones for each specific region.
Correct. You may be able to answer this, but there is a question about lower limit of cell number for what you're doing. Do you have what the least amount of nuclei you've used for your application is?
We haven't really tested this a lot. I guess PureTarget, the recommended range of input is 2 to 4 micrograms of DNA. We've done 1 microgram of DNA and even half a microgram of DNA, and you still get coverage. You do see lower coverage. So I think the amount of input you have does drive coverage. And so it's hard to say what that is. Probably, you're probably in the hundreds of thousands of cells range for that. But we haven't really pushed on what's the bare minimum we can get and still get good coverage.
Perfect. I am going through, because there's a couple of questions that I know my team can answer, but while I have you here, I think that covers specific questions for you, except we did have somebody ask, since we don't make animal models, someone was curious if you could purchase engineered mouse models generated at the embryo stage. Do you have recommendations for folks, or do you provide that, for folks external to your university?
Yeah, we can do that for folks external. Probably the best thing would be to just reach out to me via email, and then we can talk about the specifics of your project and what exactly you need and what we could do to help you. Perfect.
And then I'm going to answer one more since we're at the top. There was a question on what was lightly fixed. And in our experiments, when we did lightly fixed, it was 1% formaldehyde for 1 minute, I believe. But some of that text or compatibility stuff should be in our tech support center, so be sure to check out that. We're at the top of the hour, so I wanted to thank Dr. Freeman for your excellent presentation today. If we didn't get to your question during the webinar, we will try and follow up with you afterwards via email. And if you still didn't hear from us, you guys are also welcome to email our tech support. We have a staff that is ready and eager to answer your questions related to Fiber-seq. So with that, I'd like to thank everybody for attending, and thank you again, Dr. Freeman, for joining us.
Thank you for having me.